Then inner to this middle lamellar, this is the primary wall, and then inner to that is secondary wall. If we will discuss it in a very comparative manner, your idea will be very much clear. So let us discuss here the primary wall and the secondary wall in a comparative account, so that your concept will be fully clear. Now this primary wall, it is present in all cells. Present in all cells, but only in mature and permanent cells. Mature and permanent cells. This is not present in all cells. This is the primary wall is the only wall layer in meristems, or you can tell meristematic tissue, parenchyma, and in leaves. That is the only layer. So here it is completely absent. In all these cases, the secondary wall is completely absent. Then the primary wall is it is thin, but it is the secondary wall is thick. The secondary wall is thick, and it is three layered. And what are the three layers? It is S1, S2, S3. These are the three layers, thick. Then it is usually elastic, but this is rigid or not elastic. This is permeable, but this is impermeable. In case of primary wall, there is less cellulose and more hemicellulose. That is usually 50% of the hemicellulose is there. But in this case, there is more cellulose and less hemicellulose. It is around 25%. Here the proteins are around 5%, but here proteins only 1%. And here it is extensible. That means it can be extended, but it is not extensible. Now the primary wall, when it grows by intussusception, while this grows by accretion. It is grows, or you can tell growth by intussusception, and here growth by accretion. So what is this intussusception and what is this accretion? Usually the primary cell wall is extensible. So when it extends, new materials are inserted within it. That is intussusception. But this secondary wall, it is rigid, it is not extensible. That is why only new materials are added on it. So that is why the thickness increases. That is known as accretion. These are some of the major differences. Then in addition to this also, you will find this gradually, as the cell matures, gradually it diminishes. Gradually it diminishes as cell matures. But in this case, with the maturity, you will find there will be deposition or there will be addition of certain substances like lignin, suberin, cutin, and even silica. So gradually we can write here no diminishing, and also we can write another point, no deposition, and here there will be depositions like lignin. So lignin is usually present in case of woody substances. There may be deposition of suberin, as in case of cork cells. There is deposition of cutin, as in case of epidermis, and there will be deposition of silica, as in most of the monocots. In most of the monocots, there will be a deposition of silica. So this green layer, this is the primary wall having these characters. Then inner to this is the secondary wall which has all these characters. So you can remember in a comparative manner. Only in few cases you will get the tertiary wall, this red line which is inner to it. That is known as tertiary wall. So I am writing the tertiary wall here. Though the tertiary wall is very rare, tertiary wall, it is usually present in case of the tracheids of gymnosperms. Tracheids of gymnosperms, usually it is present, and it is usually made up of hemicellulose and xylem. But it is present only rare cases. Then there are certain other things, that is plasmodesmata. Plasmodesmata. Now what is plasmodesmata? Usually in between two adjacent plant cells there is some cytoplasmic connection. You see, now the two cells are interconnected by means of certain cytoplasmic connections, and here there are certain tube-like structures like this through which the material, there will be exchange of material between the two cells. So this is known as plasmodesmata. So plasmodesmata is the cytoplasmic connection. Cytoplasmic connection between two adjacent cells, two adjacent plant cells, and because it makes a cytoplasmic connection, that means we can call it is symplastic. It is symplastic, and here certain tube-like structures are there. They are desmotubules. Desmotubules are one type of smooth endoplasmic reticulum. Smooth endoplasmic reticulum. They connect the two cells. So this is about the plasmodesmata. Then another thing that is present is usually pits. Now usually these pits are present only in secondary wall, only in secondary wall, not in primary wall. So that point also you can hear pits present, and here you can write pits absent. That is another point. So what are these pits? Usually pits are certain connections. Pits are present only in a secondary wall. Now suppose this is the middle lamella. Now on the both sides of the middle lamella, suppose this is the This is the primary wall. This is the primary wall and this is the primary wall on two cells. Then there will be deposition, and here also there will be primary wall that will draw lateral. Now here is then the secondary wall. Now the secondary wall is here disconnected. On both sides the secondary wall disconnected. This is one cell, that is one cell. That means a middle lamella and a primary wall is present, but the secondary wall deposition is not there. This is known as simple pit. Now, so these pits usually water moves from one cell to another cell. But in case of the another type of pit known as bordered pit, in that case what happens? Now, this is the middle lamella, then this is the primary wall. This is the primary wall on both sides. Then there will be also secondary walls. Now the primary wall and secondary walls, they will be bulged. Now see, this is simple pit, while this is known as bordered pit. This is the bordered pit. So usually these bordered pits are present in tracheids and vessels, while the simple pits, they are present in simple tissues. So these are also present only in case of cell walls. So this is about the different parts of this cell wall. Now we have to discuss the different functions. So the functions are also very important. Functions of cell wall. Now one of the very important, it is, since it is rigid, it functions just like an exoskeleton, and it provides mechanical support. It provides mechanical support against any damage, That is one of the important functions. The second important function, it gives a definite shape. It gives a definite shape to the cell. The third function is it gives protection from infection by pathogens. Then it also has a cell-to-cell interaction. Then also it acts as a barrier. Barrier to undesirable macromolecules. That means the macromolecules which are not required by the cell, they will not be allowed by this. Then the next point is it counteracts turgor pressure. When due to endosmosis the cell membrane swells, expands, the cell wall, it protects it so that the cell doesn't burst. Then the next function is since cell wall is porous and continuous, it provides apoplastic pathway for translocation of water. Translocation of water. And another important function is the cell wall determines the fate of the cell during development. That means whether a cell will be root or a leaf or flower bud, what type of cell it will give rise to, that depends upon the cell wall at the time of development only. So it determines the fate of the cell during development. That means whether it will be root hair or whether will be flower bud or whether it will be leaf, that is determined by this cell wall. So this is about the different aspects of the cell wall, the chemical structure, the physical structure and the functions. I hope now a very clear understanding about the cell wall. Wish you a very bright future. Thank you very much. God bless you.